Hello, everyone. Thank you so much, Tyler, and thank you so much, Randy and Ronnie, and all the people that made this event happen. So imagine what it would be like not being able to see, to watch your vision slowly disappear, to see the letters change from color to white, the contrast lost, to not be able to see the faces of your children and your grandchildren and your friends, to not be able to see the periphery, all the details that make our life so vibrant, and only to be able to see some fuzzy little straw in the middle. And then eventually to see those things disappear and be in a world of darkness. What I'd like you to do now is close your eyes and imagine what it would be like being blind. Studies have shown that people fear blindness more than death. Studies have shown that people fear blindness second only to cancer. Blindness is common. There are more than 7 million adults in this country who are blind, totally blind. More than 170,000 children are totally blind. And blindness comes in many flavors. There are genetic forms of blindness, such as retinitis pigmentosa, age-related macular degeneration, diabetic retinopathy. But regardless of the cause, they rob people of their vision. They make life difficult for the individual, for the family, for the health system, and society. But there's hope, and you can open your eyes. And the hope is manifest here by this handsome dog, Lancelot. Lancelot, as a puppy, was born blind due to a genetic disease that is common in Swedish Briard dogs. And Lancelot underwent a one-shot gene therapy in the year 2000, which allowed him to see. He changed from a very fearful dog that would huddle for fear of bumping into things to one that was out there playing catch, playing with his friends. And here you can see he was the biomedical representative at Congress. This was his third visit to Congress to talk about the importance of biomedical research. He won many awards. He had a cage run that was studded with plaques. He ate from drank from a silver water bowl. And he lived a long life for a Briard dog, which is a large dog, 10 years still seeing after that one shot of gene therapy. And this is what gave us hope that we could treat children. And we were, like Doris Taylor before, we were told that there's no way we're going to make this work. But this is a preview of what I'm going to tell you about. Shown here is 8-year-old Corey, the first child in the US who participated in our gene therapy study, walking through the hospital using his blind cane, holding his parents' hands because he couldn't see. And then the same boy, a year and a half later, doing clearly visual things, hammering, riding his bike to his friend's house. He could go to school and see what the teacher was writing on the whiteboard, play sports. He could read books. He basically leads the life of an ordinary 9-year-old boy. Fast forward, he's now in his 20s, graduated from school and is working now, still with vision. So what is gene therapy? Gene therapies are techniques that harness DNA or RNA to treat or prevent disease. And so it can involve any product that can be encoded by DNA or RNA. Many of you have had the Moderna or Pfizer BioNTech vaccines for COVID. That actually could be considered a gene therapy because that is based on RNA, but that is not designed for long-term expression that you would need for a disease which is lifelong, that is chronic. And so I'm going to tell you the story of developing Luxturna, which is on the left side of the screen, which is the first FDA-approved gene therapy, the first worldwide approved gene therapy that is approved for genetic disease. And since that was approved in 2017, there have been 2 more gene therapies similar to this that have been approved. So, how does one become a gene therapist? People ask me. I was really lucky to have a scientist father. He was the inventor of the first laser, the helium neon laser at Bell Labs, went on to invent 6 other lasers. and loved science, shared his science with people of all ages, and I'd often serve as a sidekick for his studies. Does anybody have a wart they want to burn off? I'd raise my hand and be up there. And I just loved hearing about science, but my interest was mainly in biology. So I went to graduate school, studied cell and developmental biology, mainly in sea urchins, and then went higher in the evolutionary tree to study mouse embryos, and was injecting DNA the time when the molecular biologic techniques developed that allowed cloning and splicing genes. And the idea hit me, like many others probably, wow, maybe we can use these genes to cure or treat disease. And I searched up who was interested in doing this, and I found one investigator at the National Institutes of Health, French Anderson. I went and visited him and said, how can I become a gene therapist? He hadn't yet done a gene therapy trial, but he ended up doing the first approved gene therapy trial. And he said, oh, it's simple. Go to medical school, go to Harvard Medical School, learn about the diseases, and go back to the lab and start working on them. Okay, it's simple. Well, I applied, somehow I got in. In there, I met my husband and learned about the diseases. And as fourth-year students, Al asked me, in the glow of our first successful gene transfer experiment, the birth of our daughter, do you think we could transfer genes to the retina? And I went, sure, why not? So we went ahead and did that. But what I didn't tell him was there were so many things we didn't know. In fact, because we were so naive, I think it benefited us, because otherwise we would have bailed early in the process. We didn't know any of the genes which caused blindness. We had no animal models. We knew nothing about the natural history of the disease, no ways of delivering DNA to the cells in the back of the retina, no surgical methods, no measures to see whether it's working, and certainly no funding. In fact, the first grant I wrote to NIH was rejected, saying this is not going to work. But we slowly developed the tools. DNA is highly charged, so you can't just pass it across a cell membrane. You have to play tricks on it. And one trick is to actually take a recombinant virus that doesn't cause disease on its own, remove the guts of it, replace that with a gene of interest, and then use that to piggyback the gene into cells. And we and others found that adeno-associated virus is perfect for that, especially for delivering genes to the retina and the nervous system. And that's what we and many other people use now. As far as the genes, we benefited from the development of the Human Genome Project, where the first genes were identified around 1990, where there are now more than 280 different genes, which, if mutated, can cause retinal disease. And one of the early ones was a gene called RPE65. It was first identified in humans with congenital blindness, and then tested in a dog model that happens to be the dog model that was at our facility. And lo and behold, that dog was found to have blindness due to that gene. So we were all set. We had the dog model, we had human patients. We knew a lot about the disease because somebody had studied RP65 and shown that it's important for delivering the vitamin A precursor to retinal cells, the one that we need to allow vision to occur. And so we began our studies. And shown here, you can see this child with wobbly eye movements that are very similar to the wobbly eye movements of the dog. And they have wobbly eye movements because the retina's not functioning, not fixing and looking at a face or an object. Instead, it wobbles around. So this is the vector. I showed you that we can incorporate the gene of interest, which here is RP65. And then the next step is surgical, and that's where my husband perfected a technique to deliver the genes to the cells in the retina that need it. And you can see the cartoon here, which shows that the material's delivered through a little blister-like uprising under the retina, where the virus is exposed only to the cells that need it. And there were a number of other techniques that steps we took, making sure that the virus was clean and making sure it didn't bind to reagents we were giving to the appropriate dose and so forth. But let me show you how this manifested in the dogs. So before injection, you can see this dog is walking around bumping into objects because he can't see them. And then after treatment, the dog is walking around avoiding objects and much happier. And you can see here in this dog, which the eye was not injected, you shine a light in the eye, the pupil does not constrict, whereas after it's injected, it constricts rapidly, showing that the retina is sending signal to the brain, and the brain is sending signal back to the iris to constrict. And look, the dogs can read. No, not really. That's Lancelot reading his paper. So we were all set to go to enroll adult humans, and then disaster happened in that there were several high-profile gene therapy problems. The major one was a death in a gene therapy trial that was unrelated to our study, using a totally different vector, using systemic delivery of orders of magnitude more reagent than we would ever think of using for the retina. And the whole field came to a screeching halt. There were Senate investigations, and all the funding dried up of gene therapy, and nobody wanted to be involved in gene therapy. So here we had our best results, no place to go. But thankfully, Catherine High, who is a hematologist at the Children's Hospital of Philadelphia, CHOP, had been wise and hired all the people who were being fired and got funds from the hospital to run 2 gene therapy clinical trials, one in her disease, which is hemophilia. And she asked me one day, Jean, how'd you like to run a clinical trial for blindness? I'm like, of course. So we were off to the races. And so new issues for a research scientist, learning all of these acronyms and all the paths to drug approval, the different types of trials you go through, the regulatory issues. And again, the regulatory issues were really challenging because of the gene therapy problems that I just mentioned. So we got to the point of going through all those hurdles, and then we had 2 steps which were somewhat of a challenge. One, no children had been enrolled in clinical trials for non-lethal diseases. So we had to be grilled by what was called the NIH Recombinant DNA Advisory Committee, aptly named the RAC. Where fortunately there were patients and families who came and spoke about how difficult it was to raise a blind child. The whole committee voted unanimously to allow us to proceed, and now children can be enrolled in these clinical trials. And then most affected individuals in the United States hadn't been genotyped because there was no treatment. And so we were fortunate to be able to collaborate with a group in Italy who had the wisdom to genotype their patients, and these patients were readily available. So our first 4 patients were Italian. So this shows the subretinal injection procedure that my husband developed using a needle that is the width of an eyelash to deliver this material. You can see this localized retinal detachment, which then exposes the cells that need fixing in the retina. It flattens down within hours, leaving minimal evidence that any surgical procedure has been done there, and allows those cells to take up the gene in residence and start producing the protein that it encodes. So we're ready to enroll patients. But there were some funny things that happened. We didn't know what the real risks. We knew that from all of our safety studies, which were meticulous, that there was a very minimal safety risk and very high efficacy prospect of benefit. But there was the unknown. Nobody had done this before. So one thing that we thought of, well, maybe people won't like their vision. Maybe we'll have to tell them there's an exit strategy. And so the consent form said, if you don't like your vision, we have ways to remove it. Nobody chose that route. Also, because it wasn't clear whether or not the material would expose the rest of the body, and it doesn't, we had to tell all of the people in our study, including 8-year-old boys, you cannot father a child for a year after the study. And that caused some humor in the consent process, So this shows that boy that I showed earlier on. using his untreated eye, walking the maze, very similar to what we had the dogs do to walk through and avoid obstacles. And you can see that he immediately went off course and bumped into obstacles, had to be re-guided, whereas now he's using his injected eye, following the course accurately, avoiding obstacles to the side. He's stepping over an obstacle in his path and finding his way to the end. So then we had a few other challenges. We had showed that this was effective in all the people involved in this phase 1 study. We went to the FDA. We'd like to propose to go through a phase 3 trial to get this approved as a drug. And they said, you must inject the second eye. And that was a concern to us because nobody had successfully re-administered a gene therapy reagent because of fear of immune response and so forth. And also you have to include a control group because what if people get better on their own? Well, we knew that didn't happen, but we didn't have the data to show it. And we also worried that if people were assigned to a control group not to get treatment, that nobody would enroll because the word had gone out that this was effective treatment. So we went back to our trusty animals and injected one eye high dose, the second eye high dose at the time when immune response would be maximal. You can see Venus here is very visual, no problems with re-administration. And after these experiments were over, these dogs came to live in our house because we grew so fond of them and wanted to give them life outside of the colony to enjoy grass and chasing squirrels and so forth. As far as the trial design, we developed a trial where people were assigned randomly to intervention versus control, but the control group after 1 year could cross over and have both eyes injected and then be followed for 1 year to the end point, which was performance on a mobility test, which I'll get to in a second. That way, everybody would be assured to be able to a prospect of benefit. But then the challenge was that the FDA had only one measure that they would approve a new drug with for eye drops, for example, and that was improving 3 lines on an eye chart. Well, many of our patients could not even see an eye chart. That was not going to work. So we had to propose another clinically meaningful measure, and that we decided based on our results in the dogs and the exploratory obstacle course measure to develop a mobility test which we've then validated. We designed multiple templates to avoid learning effect and use this as our primary outcome measure. And shown here are results of one patient, shown at baseline and then afterwards, 2 years later. You can see at the baseline, this young man goes off the course immediately, has to be guided onto the course, whereas at the 2-year point, he follows the arrows perfectly and avoids all the obstacles and finds the end of the course really rapidly. And the results of the clinical trial were outstanding and led to approval of the drug. Now, how did this affect one individual? And I'm going to talk over this. This is Christian Gardino, who went public with his story during the trial. He did not learn how to sing from gene therapy, but in fact, he went to the Apollo Theater as a kid to sing. That's in New York. And would be led up the stairs onto the stage, would stand still and not be able to see anybody in the audience, and would not move for fear of falling off the stage or tripping over wires or something. And after the gene therapy, he would get on stage, dance, see people's faces, as you can see here, become independent, and shown here was his performance last year on American Idol. He came very close to winning American Idol. He even ran out on the catwalk, which made us worry, oh no, we don't want him to fall off, but no problem at all visually. And he made Katy Perry cry. I'd like also to tell the story of another remarkable woman, Caitlin Corey Davis, who was one of the first people to enroll in our trial as a child, who tells her story at the US FDA. I'll stop talking in a second. I want to share this story because Caitlin participated in our study, benefited immensely from gene therapy, and very sadly, years after the gene therapy, died last year of an unrelated disease. And her parents want her pioneering spirit to be spread. So it can be louder.
At first, vision loss is just small things going. When I could no longer see pencil on paper, I used Sharpie. Text is too small, I got large print books. Having trouble with colors, who cares? I wear a uniform. But I don't need to tell you about the natural history of the disease. Vision loss while in school is like the old Lewis Carroll quote from Alice in Wonderland. My dear, here we must run as fast as we can, just to stay in place. And if you wish to go anywhere, you must run twice as fast as that. Spending all this time and energy attempting to adapt to my life with ever-dwindling vision did not leave much room to live. I was at the precipice of losing it all. I knew I could eventually adapt to being a blind person, but my passions for math and science may not be realized, and that was devastating. It would have been one thing if I had lost my vision after completing my education. At least then I would have had the knowledge. But this meant that even though I could keep running, I could no longer stay in place, but would begin to fall behind. But that very November, the phase 3 clinical trial for Veretagine, I was finally treated with the gene therapy in December of 2013, days before my 21st birthday. And let me just say, that was the best birthday I ever had. I was well aware of the risks and benefits that this treatment entailed, and I would do it over and over again. Within days of the first surgery, I could see vibrant colors again. I was no longer living in a black and white film. I could see the clock tower of Philadelphia City Hall, sculptures, windows and all, at night, when mere days before, I thought it was the moon. I could walk confidently in dimly lit settings, indoors and outdoors. And then there was the sun. It seems funny to say now, but man, that thing is bright. I could practically feel my pupils contracting, which my owl-like eyes would never do before. And then I could go to a restaurant on my birthday, see the plate, the utensils, glasses, and people at other tables, all by candlelight. These changes were just the beginning. I could use adaptive technology, the iPhone accessibility apps, Zoom features, and more. I was independent and mobile, which I had not been for some time. I've gained normal vision, but I gained all of my independence. I just want you to know that this was significant to me, significant in the way that I live and plan my life. I no longer had the fear of what the next year would take away from me. I went to graduate school and got my master's in science of epidemiology this past June. I finally can live my life the way I want to.
Remarkable young woman, and we miss her. So Veretagine was, Naparvovec Rizal was approved in 2017. Amazingly, we had applied for use age 4 years old and over, but the FDA and EMA were so impressed by the results that they approved it for children 1 year old and over. And there was a race in the US for the first patient to be prescribed this drug. Shown here is Monroe, 4 years old, who was taken out for Halloween after her first injection, and she was not interested in candy at all. She was interested in the fact that she could run around in the dark and see things. And there's Creed, who achieved his lifelong dream of throwing away his blind cane, and Jack at Harvard, who was more cerebral, talking about what he could see at the whiteboard and riding his bike. But it's been a huge eye for gene therapy, and you don't want to be the last gene therapy. Importantly, there are now more than 40 different clinical trials for different forms of eye disease that are listed here, including age-related macular degeneration and diabetic retinopathy, that are many of which are in late-stage trials with very promising data. There are multiple strategies being used, and I've listed those on the right, but importantly, one of them is using gene editing. This is the first gene editing gene therapy that's delivered directly to a person. And David Liu, who is speaking later today, was the founder of the company that started that trial. And gene therapy is now in multiple countries, continents, and places around the world. I'd like to sum up by saying there's now a path for delivering gene therapy for inherited disease, not just retina, but systemic disease in the United States and worldwide, that these vectors are now in pharmacies, operating rooms, and in patients all over the world. And it's being used now to evaluate common retinal diseases. Like all good science, many of the learnings will ultimately be applied to studies that are far removed from the original ones. And I'd like to acknowledge all of the participants in these studies who are the real pioneers, including the dogs who led the way, and also note that I am just the spokesman for an incredible team of very talented and complementary investigators listed here. Thank you very much. Thank you.